All right, hello everybody. On Zoom, I can't really see you at the moment, but can you all put into one of you put into the chat whether you can hear me? Yay, okay, thank you. And hopefully you can see the presentation that's up. Okay, perfect. All right. Okay, so for in-person and Zoom folks, thank you very much for being here. We have a presenter today, but we also have somebody to introduce our presenter to you. So Christine, I'm just going to hand it right over to you.
Thanks so much. Yeah, welcome everyone. It's my pleasure today to introduce Sam Zlotnik. I am Christine Miller and I'm Sam's PhD advisor, and I'm zooming in from the UK. So it's getting a little dark here, and hopefully you can all hear me okay. So I'm excited to introduce Sam for this important event. Sam completed his undergraduate degree at Berkeley in biology and psychology. During this time, he worked as a field tech for the Nature Conservancy and for other organizations. Sam later completed his master's degree at Purdue University, working with Jimena Bernal. There he studied behavioral ecology using toads, the cane toad and the American toad. We then convinced Sam to work with some insects here when he moved to Gainesville and to complete a PhD in SNRE with a concentration, and he did a concentration in statistics while he was here. And also during his time here, he TA'd an environmental science lecture and lab course, which involved planning field trips, which he enjoyed, and he's told me that he learned quite a bit from that experience. In recent years, Sam became interested in science writing and received a AAAS Mass Media Fellowship. So last summer, Sam was working at the Smithsonian Magazine as an intern, which sounded like quite an amazing experience. And so when Sam completes his PhD, he will be pursuing a career as a professional science writer. It's been a real pleasure to work with Sam, and without any further delay, please welcome Sam Zlotnik.
Great. Well, thank you so much, Christine, for that lovely introduction. Can everyone hear me all right? Perfect. So yes, thank you all for coming. And today I'll be telling you a bit about my dissertation research, which is on the feeding behavior and mouthpart development in juvenile leaf-footed bugs. So I'd like to start with talking about juvenile animals a bit more broadly. So in many animal species, juveniles are faced with heightened dangers in their environment, as well as challenges in acquiring the resources needed to grow and develop into adults. So for example, these sea turtle hatchlings you see here, they face a notoriously hazardous journey just getting from their nest to the ocean for the first time. And studying juvenile animals and how they're impacted by these challenges is really critical, especially for understanding and predicting larger scale population dynamics. So in some threatened and endangered species like the stellar sea lion and the bog turtle, which are both pictured here, mortality in the juvenile stage is a major factor determining population level trends. So considering sources of mortality and survival adaptations in juveniles rather than just adults is super important. And among the many challenges that juveniles face, feeding may be especially difficult. So juveniles often have underdeveloped morphology and are less experienced foragers compared to adults, which means they may be weaker competitors while also being at a higher risk of predation while foraging. So an example of this is in these European shags, such as this bird pictured here, and juveniles actually spend more time foraging than adults just because they're less proficient at it. So because of these feeding challenges, juveniles may have a harder time than adults accessing the nutrition they desperately need to grow and develop. And when they do manage to feed successfully, they may rely on different feeding strategies than adults. So even in species that eat the same food throughout their lifespan, juveniles may be using slightly different strategies than adults. However, research into feeding adaptations in animals often focuses on adults rather than juveniles. So the feeding strategies used by juvenile animals in particular, as well as their limitations, are often less well understood. So this leads me to two big questions, which I addressed in my dissertation. First, what feeding strategies do juvenile animals rely on to feed? And second, what are their limitations? So I'm going to be returning to these questions in a minute, but first I want to talk in a bit more detail about what kinds of feeding challenges juvenile animals might face. So juvenile animals may have an especially hard time if their food can defend itself. So of course, prey animals have many physical, chemical, and behavioral defenses. Like this armadillo has hard armor and it can roll in a ball, so it's very well defended. A scorpion can inject venom with its stinger. So there's many different types of defenses that we're pretty familiar with in animals. But defenses aren't limited to animals. So juvenile herbivores may face difficulty feeding due to the defenses used by their host plants to deter herbivory. So for example, many plants produce chemical defenses like the coffee plant shown here on the left, and on the right is deadly nightshade. So coffee plants obviously produce caffeine, and caffeine actually has insecticidal properties. And nightshade has a whole bunch of toxic compounds, including atropine. So there's been a good bit of research done on the adaptations that help herbivores feed on chemically defended plants. That's like a big area of study. But there's been less attention paid to structural defenses. Structural defenses can include a whole range of things, including some examples here: shells, spines, and latex. These defenses may pose challenges to juvenile herbivores because of their smaller body size and the smaller size of their feeding-related morphology, like their mouth parts, claws, and other appendages. So I said I'd be returning to these questions. I've kind of narrowed things down a little bit. So for my dissertation research, I asked, what feeding strategies do juvenile herbivores rely on to surpass structural plant defenses? And secondly, again, what are their limitations? And in particular, I focused on two potential feeding strategies that juvenile herbivores may use. The first is developmental plasticity and feeding-related morphology. And the second is feeding facilitation by adult conspecifics. So as I'll explain in a minute, both of these strategies are especially likely to be useful to juvenile animals. And neither strategy has been super well studied across a range of taxa or environmental contexts. So of course, there are many, many feeding strategies out there. These are just two that I identified that were relatively understudied. So first, I'm going to talk a little bit about developmental plasticity, and specifically plasticity in feeding-related morphology and what I mean by that. So in some animals, the development of mouth parts or other feeding-related morphological traits is actually determined or driven by the juvenile diet. So the size and shape of those mouth parts or other traits are actually to some degree optimized for that particular diet. So an example of this comes from these spade foots, which are these frogs you see at the top. And as tadpoles, they can develop into different morphs. So either the majority of them become omnivore morphs and they eat plant matter, as well as some animals. But then there's this alternative carnivore morph, and they only develop into carnivores if they consume enough shrimp. And they actually get larger jaws, so they're better at eating that animal-based diet. And similarly, juvenile rabbits will actually grow denser teeth after feeding on tougher plant matter. So that makes them better able to feed on other plants that are tougher. So as these examples illustrate, developmental plasticity in mouth parts and other feeding related traits, they may be an adaptation used by juvenile animals with a variety of diets. It's definitely not specific to herbivores. But for juvenile herbivores feeding on host plants or plants with structural defenses, this kind of plasticity holds great potential as a feeding adaptation, as it could enable them to develop morphology that is to some degree optimized for overcoming these specific physical barriers, similar to the rabbit example. And the second feeding strategy I investigated was feeding facilitation by adult conspecifics. So of course, some juvenile animals are fed by their parents. Their parents just like give them food. But in many animal taxa, they don't have this form of parental care. However, juveniles may still benefit from feeding near unrelated adult conspecifics if feeding by adults causes damage to physical barriers, such as structural defenses in plants. I just realized I didn't define conspecific as just another individual of the same species for anyone who's not familiar with that term. So an example of this feeding facilitation is in soapberry bugs. Those are these bugs here. And in these bugs, they feed on multiple different types of seeds that have shells. And the juveniles will actually have higher survival if they feed on a seed that has been previously fed on by an adult. So the likely explanation for this is that the adults will drill through the shell in the process of their own feeding. They will get through this shell with their own mouth parts to feed on the seed inside. And then if a small juvenile comes by and might struggle to get through that shell on its own, it can just reuse the hole that was already made by an adult. So evidence for this explanation comes in the form of a series of experiments that showed that older juveniles have less of a benefit from adults, likely because they're just better at getting through the shell on their own. There was also less of a benefit of having an adult feed first if the shell was already cracked, or if they were feeding on a type of plant that had thinner shells. So the shell was just easy to get through. So the juveniles really only got this benefit if they were very young juveniles feeding on a really hard shell that was uncracked. In that case, having an adult feed first made it easier for the juveniles to feed after them. These findings provide strong evidence that feeding by larger conspecifics, i.e. adults, enables young juveniles to more easily surmount the plant's structural defenses. So these are the two potential feeding strategies that I investigated in my dissertation. And in particular, I was interested in whether or not these strategies are used by juvenile herbivores, and if so, how effective they are at helping the juveniles feed successfully on structurally defended host plants. And the herbivores that I used to investigate these feeding strategies were leaf-footed bugs, which are true bugs in the family Coreidae. And leaf-footed bugs are highly useful for studying how plant structural defenses limit herbivory, because they feed on a wide range of different types of plants and different parts of the plants, and that includes fruits and seeds with thick shells, rinds, skins, etc., and other types of physical barriers to feeding. I feel like I should explain the mouth parts a little bit because that's going to be key here. These bugs have piercing, sucking mouth parts, which are all the mouth parts together collectively called a rostrum. So I usually just refer to it as rostrum. But the full rostrum actually includes an outer sheath, which is called the labium. And that surrounds this bundle of 4 sort of thin probes that are called stylets. The CT scanned image on the left shows these the labium surrounding the inner stylets. And the stylets actually form sort of like a straw-like shape. It's kind of like a multi-part straw, but you can just think of it as a straw, and it can be inserted into the food item. It actually shoots out some liquid with digestive enzymes, partially digests the food, makes sort of like a soup inside the plant, and then sucks up those nutrients. And on the right, the image on the right, you can see a leaf-footed bug feeding on a tomato. And here, so the labium has 4 different segments, and the first 2 segments are bent back in this image. So it kind of partially exposes the stylets. You can see that super thin line underneath the bug's head. Those are the stylets. And as the labium is being bent back, the end of the stylets are being inserted into the food. So here, this bug is just barely inserting its stylets into this tomato. But we have actually seen these bugs bend back their labium in its entirety and fully insert their stylets into the food. So they actually have their face like pressed up against the tomato or whatever they're feeding on, and their stylets are fully into the food, which means that the length of their rostrum is the key factor determining how deep they can penetrate into that food item. And the juvenile bugs, which are called nymphs, they're similar to adults in their overall morphology, but they're smaller in size, and because of that, they have a shorter rostrum that can't penetrate as deep into the food. So in this photo on the left, you see an adult and a nymph of the same species, and I've highlighted the length of their rostrum in this like lime green line here. You can see that the nymphs have a shorter rostrum compared to the adults. And in this photo on the right, you can see nymphs of various different developmental stages, which are called instars. They have 5 different instars before they become adults. And you can see that their size varies a lot, going from first instars through 5th instars. And their rostrum's length obviously varies as well. So the smaller overall size and shorter rostrum of nymphs compared to adults may limit nymphs' ability to feed through barriers such as the shells of nuts or the rinds of fruits. And these bugs don't have fully developed functional wings until adulthood. So that's key because nymphs are limited in their ability to disperse to find new host plants. So if their eggs were laid on a certain plant that is really well defended, and then the nymphs really struggle to feed, they can't just go fly and find another plant. They're kind of stuck with whatever is in their immediate vicinity. So for all these reasons, leaf-footed bugs are a great taxon for studying these two feeding strategies I talked about earlier. And there also have been a couple of recent studies that provide preliminary evidence for both of these strategies in leaf-footed bugs. So I'm going to go over these recent studies briefly. Okay, so first, there's been a recent study showing that a species of leaf-footed bug that feeds on cactus fruit will grow a longer rostrum relative to their body size when raised on fruits with thicker skins. So in this study, these bugs were collected. Populations of the bugs were collected in Florida and Texas, and then they also collected species of cactus from Florida and a species of cactus from Texas. The Texas cactus fruit was bigger and had thicker skin than the Florida cactus fruit. And bugs from both populations, when they were raised on the Texas fruit, they actually grew a longer rostrum relative to their body size than when they were raised on the Florida fruit. And so that was true for both populations of bugs. This study was the first to show evidence of rostrum length plasticity in leaf-footed bugs. But since the fruits actually came from different cactus species, there is still the possibility that other factors besides the fruit skin thickness, such as maybe nutritional content or other characteristics of the fruit, might have also differed between them and contributed to the differences that we're seeing here. So there's still more research that needs to be done to really show this definitively, that it was due to the skin thickness in particular. And in another species of leaf-footed bug, this species feeds on conifer seeds, and in this experiment they use pine nuts, and it provides a potential example of feeding facilitation. So similar to the soapberry bugs, these bugs have to drill through the shell of the seed to get to the seed inside, which is the part they actually eat. And in the image on the left, you can see a pine nut that's been fed on by two different bugs. And the seed inside is all like shriveled up because it's been fed on extensively. And the arrow points to the single feeding hole in the shell. So the first bug came, fed through the shell, drilled through the shell to get to the seed inside. Then the second bug came and just reused that same hole. There was no other hole in the shell. So this provides some evidence of feeding facilitation, potentially in this species. But this is crucially between two adult bugs, and it's still unclear whether nymphs would also reuse feeding sites or whether they can actually improve their ability to access nutrients by reusing adult feeding sites in particular. So the benefits of reusing the hole in the shell were not directly tested. So for my dissertation research, I had 5 main aims. First, I set out to test the extent of rostrum length plasticity in leaf-footed bugs and its ability to help nymphs survive on structurally defended plant diets. So in particular, I wanted to determine whether the plasticity documented earlier in response to feeding on fruits from different cactus species could actually be attributed specifically to the thickness of a feeding barrier rather than to other diet-based differences such as nutrition or potentially other characteristics. And my second aim was to examine whether adults can improve nymph survival when nymphs are raised on a diet of seeds with hard shells. So this aim tests whether feeding facilitation can actually help nymphs survive on host plants with structural defenses that are likely to pose a particular challenge to nymphs because of their small body and rostrum size. And then with my third aim, I wanted to look more specifically at the nymph's behavior. So here I investigated conspecific attraction in nymphs. And I figured that if juvenile leaf-footed bugs have an adaptive strategy of reusing adult feeding sites to gain nutritional benefits, then they would also presumably generally show some sort of attraction to conspecifics in general or adult conspecifics in particular. And so I set out to test whether nymphs showed this type of attraction behavior specifically in a feeding context. And my fourth aim was to specifically determine whether or not nymphs seek out and reuse adult feeding sites. So I thought this was key to actually see if the nymphs are going and using those same sites, because that would be a necessary thing for them to benefit from feeding facilitation. And then my fifth aim involved directly testing what impacts, if any, adults have on the growth and survival of nymphs. So Even if I did find that nymphs showed attraction to conspecifics and reuse their feeding sites, it wouldn't definitively demonstrate the adults are actually providing any sort of nutritional benefits or other adaptive benefits to those nymphs. So I thought measuring fitness benefits directly was an important step in investigating the feeding facilitation hypothesis. So these are the 5 aims, and I'm going to be starting off with number 1, which is to test the extent of developmental plasticity in rostrum length. So for this project, I used a species of leaf-footed bug called Leptoglossus zonatus, which has an extremely broad diet. It feeds on at least 24 different families of host plants and mostly consumes the fruits and seeds of those plants. Here it can be seen feeding on tomatoes and snap peas. And across this broad diet, there are many host plants that have structural defenses protecting their fruits and seeds. So here they're feeding on tomatoes, which aren't really well defended, but they also feed on a bunch of nuts and citrus, which often has thick rinds. So depending on where these bugs live during their early development, they may face thick physical barriers that they would need to overcome in order to get the nutrition they need to survive to adulthood. So because of the species broad diet, we were actually able to make a semi-artificial food type to feed them. So we made these agar plates with raw sunflower seeds embedded under the surface. And I call it like a semi-artificial diet because the sunflower seeds are a natural part of these bugs' diet, but agar is obviously not. So they don't actually feed on the agar, they're just feeding on the seeds. And the agar is really just sort of a stand-in for a physical barrier or structural defense, such as like the rind of a citrus fruit. So there are 2 different diet treatments that we used. The thin barrier treatment that had a very, very thin layer of agar covering the seeds. So it was just like barely any agar on top of them. And then the thick barrier diet had a 10 millimeter thick layer of agar covering the seeds. Overall, there was the same amount of agar and the same amount of seeds in each treatment. It's just how thick was the barrier covering them. That was the only thing that we manipulated. So these diets allowed us to change the thickness of the physical barrier to feeding without actually altering the quality or quantity of nutrition provided to the bugs. And leaf-footed bugs go through 5 developmental stages called instars, like I mentioned before. And they don't actually begin to feed until their second instars. So for our experiment, we actually raised bugs in communal rearing cages until they were 4th instars and then put them into these 2 treatment into containers with one of the 2 treatments, either the thin barrier diet or the thick barrier diet. And the bugs remained in the treatment containers throughout their 4th and 5th instars. And once they had closed as adults, we froze them and photographed them to take precise morphological measurements. And the 2 most important measurements we took were the pronotum width and the rostrum length. Pronotum width is a commonly used proxy for body size. So from now on, I'm just going to refer to it as body size, but you can see what it looks like here if you're interested. So getting into the results, first I'm going to show the results for body size alone with bugs raised on the thin barrier diet in dark blue and bugs raised on the thick barrier diet in yellow. And I separated out the male and female bugs because female bugs tend to be larger on average than male bugs. And as you can see, the bugs raised on the thick barrier diet grew up to have smaller body sizes at adulthood than the bugs raised on the thin barrier diet. So this was true for both the female bugs and the male bugs. And then if we looked at survival, the bugs from the thick barrier diet also died at higher rates. So they had lower survival than those from the thin barrier diet. So overall, based on both growth and survival, we can say the agar barrier definitely limited growth and survival of these bugs. And next, I'm going to show a scatter plot similar to the one from that the cactus fruit study I talked about earlier. So this shows the bugs' rostrum lengths relative to their body size. Body size is on the x-axis and rostrum length is on the y-axis. And here, again, blue refers to thin barrier, yellow refers to thick barrier. And then I also added this gray dashed line, which shows the thickness of the thick barrier. And here are the results. So as expected, we found the bugs raised on the thick barrier diet grew longer rostrums relative to their body size than those on the thin barrier. And this difference between treatments extended across the whole range of body sizes. So even these much larger bugs with really long rostrums that were long enough to feed easily on the sunflower seeds, they still showed this effect. So you can see the yellow line is like shifted up compared to the blue line. It has a higher intercept, but their slopes were similar. So this effect of the barrier seemed to be consistent across body sizes. And overall, this provided good evidence that the bugs have plasticity in the growth of their rostrums as a direct response to the thickness of the physical barriers they're feeding through. So to return to the original aim of this project, it was to test the extent of developmental plasticity in rostrum length. And the study provided the first evidence of rostrum length plasticity in leaf-footed bugs that can be specifically attributed to the thickness of the physical barriers rather than to other characteristics of the diets. And based on this finding, we can conclude that mouthpart plasticity may potentially facilitate feeding and be an important factor in maintaining diet breadth in Leptoglossus sonatus. But we also found that physical barriers surrounding fruits and seeds can limit the survival of even highly generalist feeding species like Leptoglossus sonatus. So while plasticity may help these nymphs feed more effectively, they're still limited in their ability to overcome these barriers, such as those imposed by structural defenses in their host plants. So to put that more succinctly, we can conclude that nymphs have mouthpart plasticity, but still suffer a higher mortality when faced with barriers to feeding. So there are limitations to the strategy. So now I'm going to be moving on to the second aim. So we're going to switch gears from talking about plasticity to talking about feeding facilitation, which is what I'm going to be talking about for the rest of the presentation pretty much. So for this aim, we examined whether adults can help nymphs survive when feeding on nuts with hard shells. And in this experiment, we use the same species of leaf-footed bug, Leptoglossus sonatus, but this time we're feeding them a pecan-only diet rather than this sunflower seed and agar diet. And these bugs are known to feed on pecans throughout their lifespan, but we still thought that nymphs might have a harder time feeding through the hard pecan shell than adults, just because they're so small and have such a small rostrum. So we hypothesized that because of potential feeding facilitation from adults, if adults made a hole through the pecan shell, then nymphs would have an easier time feeding. And so the presence of an adult would kind of ameliorate this, this feeding challenge imposed by the pecan shell. So to test this, we took newly emerged second instars and assigned them to one of 4 treatments, which are illustrated here. As a reminder, the bugs don't begin to feed until their second instars. So essentially in this experiment, as soon as the bugs started feeding, we put them in the experimental treatments and they stayed on these diets throughout their juvenile development. And so in the treatment cups, each focal bug was given 1 pecan, either with or without a shell. And they got a conspecific bug, sort of like as a neighbor, which was either an adult or another nymph, also a second instar. So throughout the juvenile development of the focal bug, if the conspecific died, we replaced it with either another adult, if it was an adult, or with another nymph, if it was a nymph, and we matched the developmental stage of nymphs so that the focal and non-focal bugs or non-focal nymphs were always of the same instar. And then if the focal bug died, we just recorded how long it had survived. So for the results of this experiment, I'm going to show it on a survival plot, which shows each of the 4 treatments in a different colored curve. The x-axis has the number of days past the start of the experimental treatment, which again began as soon as they became second instars. And the y-axis shows the proportion of bugs surviving, which can also be thought of as survival probability. And before I show you the actual results, I'm going to show you what we expected to see based on the feeding facilitation hypothesis. So these are the expected results. The top 2 lines that are shown here in blue and this dashed green depict the high expected survival of bugs in the treatments without pecan shells. So without these defenses, we expected bugs to pretty much just do fine and get plenty of food. Of course, there would be some mortality due to other factors, but we expected the bugs to have relatively high survival. And we also didn't expect that the life stage of the conspecific bug would have any effect on survival of the focal bug. In contrast, the red and orange lines are lower than the blue and green ones, because those showed the treatments with the pecan shell. And we thought that with the shell, the nymphs would struggle to feed, especially like those early instars. So like the second and third instar nymphs would really struggle to feed through the pecan shell, and so would have lower survival. But the orange line is higher than the red line because of that feeding facilitation hypothesis. So the orange line shows the shell, pecan with the shell plus an adult conspecific. And so we figured if an adult was there feeding on that pecan, it would create holes in the shell. And then the nymph would be able to feed through those same holes and access those nutrients more easily than the nymphs in the treatments with pecan shells plus a nymph conspecific. So these are the expected results. And now I'm going to show you what we actually found. So as you can see, there's some similarities and some differences from our predictions. We did find that pecan shells significantly decreased the focal bug survival, especially at the earliest developmental stages. So again, like second or third instars. So the pecan shells definitely induced high mortality in those second and third instars, which have very small body sizes and short rostrum lengths compared to adults. You can see that the slopes of the orange and red lines from like day 0 to 10 are much steeper downward than the slopes of the blue and green lines. However, the presence of an adult rather than a fellow juvenile conspecific did not significantly affect the focal bug survival. So here you can see the blue and green lines, the confidence intervals are overlapping a ton, and the orange and red lines, confidence intervals, again, overlapping a lot. So that shows the difference between, or the lack of differences between adult versus nymph non-focal bug, both without a shell and with a shell. There was no real effect of the conspecific. So based on this, we can say feeding facilitation by adults did not seem to help the nymphs feed through the pecan shells. So to review, the main aim for this project was to examine whether adults can improve nymph survival when feeding through hard shells. And contrary to our predictions, we found no evidence that feeding facilitation by adult conspecifics really helps Leptoglossosonatus nymphs feed through physical barriers like nutshells. And again, we saw that these feeding barriers, which are often part of host plant structural defenses, can really limit the survival of even species like Leptoglossosonatus that is a highly generalist feeding herbivore. So again, that points to really looking at this juvenile period and you think about what these bugs can be feeding on. So overall, we can conclude that nutshells limit nymph survival and adults do not help them surpass the feeding barrier. So while we didn't find any evidence of feeding facilitation helping Leptoglossosonatus nymphs feed through pecan shells, we also wanted to test this feeding strategy in a more specialist feeder as well, as the previous evidence for feeding facilitation came from soapberry bugs and the conifer seed bug, like I talked about earlier. And both of them primarily feed on seeds with shells. So we thought juveniles might be more likely to evolve behaviors to take advantage of feeding facilitation in more specialist feeding bug species, since the feeding pressures are more consistent over, across populations, across generations, compared to a generalist species like Leptoglossosonatus. And since there was already some evidence of feeding facilitation in these bugs that feed on seeds with hard shells, we wanted to also test it in a species that feeds on very different food type, so without nutshells. So that takes us to Narnia femerata, which is this leaf-footed cactus bug that I talked about earlier. It feeds solely on the prickly pear cactus in the genus Opuntia. And these bugs really rely on the cactus fruit in particular. So they can get some moisture and some potentially some nutrients from the cactus pad, but they really don't do well if they don't have the fruit. So they really rely on the cactus fruits. And of course, when we think about anti-herbivore structural defenses in cactus, we think about spines because we're humans and spines stick in your fingers and they hurt. But for the bugs, the spines aren't really much of a problem. So the bugs aren't really deterred by spines, especially in terms of feeding. That is not really what we're talking about here in terms of defenses. And unlike nuts, these fruits also don't have hard shells, like I said, that could protect them from feeding by bugs. But this is also the same bug species that we had previously seen had rostrum length plasticity in response to the fruit skin thickness of the cactus fruits they fed on as juveniles. So like Leptoglossus sonatus, Narnia can grow longer rostrums relative to their body size when raised on fruits with thicker skins. So even though we know that these bugs are super well adapted to specifically feed on cactus fruits, based on these results, there's reason to expect that smaller bugs with shorter rostrums may struggle to reach the most nutritious parts of the cactus fruits, which are often the seeds. The seeds have protein and fat, which is not really found in just like the pulp of the fruits, and that's often deeper inside the fruits. And like I mentioned before, like all leaf-footed bugs, Narnia, adults are larger overall than nymphs, and they have longer rostrums. So adults may be able to reach the nutrition within the fruits, and especially those seeds, more easily than nymphs because of their longer rostrums. And then when a bug feeds on a fruit, it secretes a gel-like saliva that forms this sort of semi-permanent tunnel. It's called the salivary sheath. You can see in this photo here, they're stained in pink. So that shows where the bug has inserted its stylets into the plant. In this case, this is actually a sunflower plant stem, and it is a different species of bug, but the similar thing happens with Narnia feeding on the cactus fruit. And here the salivary sheath is going straight to the xylem. So the bugs can actually sort of navigate inside their food item to get to where they want to get it, to get to the moisture or nutrients or whatever they need to get to inside the plant tissues. So in the case of Narnia, that might be navigating towards the seeds if they need more protein. And these salivary sheaths, since the saliva kind of gels, it lasts a few days. And so if an adult feeds on a cactus fruit, then a nymph could potentially go and feed in that same site and access the nutrients more easily that it couldn't access on its own because of its shorter rostrum. So even though these cactus fruits don't have like a hard shell, like we would think of typically with structural defenses, still this feeding facilitation hypothesis could be at play, and reusing the adult feeding sites could really be important for helping them access the nutrients they need to grow and develop. So that was a whole intro to the next three aims, which all use Narnia as our study system. So here we're going to start directly examining nymphs' behavior. So for aim 3, we're investigating conspecific attraction behavior in nymphs. And of course, there are many reasons why juvenile animals might be attracted to conspecifics. You know, they could be, you know, trying to stay with the group to avoid predation, et cetera. And this feeding facilitation hypothesis is just one of them, but we thought it was important to look at, and especially in a feeding context, to look for evidence that the nymph's behavior actually aligns with this hypothesis. And in particular, we wanted to see if nymphs could actually be attracted to the cues left behind by conspecifics. So not just, oh, look, there's that bug over there feeding on the fruit, I'm gonna go, you know, hang out near that other bug, but can the nymphs detect where another bug has been feeding previously and go towards that fruit that another bug has already fed on? So to investigate this conspecific attraction in Narnia femerata, we first collected cactus fruits and size matched them in pairs. Then one fruit from each pair we put in a container with a bug for 1 week, so it was fed on by that bug. And the other fruit was left in an identical container in the same room right next to them, but with no contact with any bugs. And after the end of the week, we started a 3-day behavioral experiment. So both fruits from each pair were stood upright inside a larger cup. And then we put one nymph into each cup and recorded its location every 10 minutes for 10 hours a day for 3 days in a row. And each observation was pretty much just writing down where each nymph was in the cup. So was it on the fruit, not on the fruit? If it was on the fruit, which fruit was it on? And we repeated the same experiment twice. So in the first trial, the fruits were previously fed on by an adult bug. So we were testing whether nymphs were attracted to feeding cues left behind by adult conspecifics. So that's, we call this the adult fed trial. And then in the second trial, the fruits were fed on by other nymphs. These are all second or third instar nymphs. So this time we tested nymphs' attraction to feeding cues left by other unrelated nymphs. And so this is called the Nymphed Trial. And after completing these experiments, both trials, we counted the number of times that each nymph was observed on the treated fruit and subtracted the number of times that each nymph was observed on the control fruit. And then with these differences in observations, we averaged them across all the nymphs in the experiment. And that gave us a measure of the overall attraction to or avoidance of the treated fruit relative to the control fruit. And so a positive value of this measure would indicate a preference for the treated fruit on average. And a negative value would indicate there was a preference on average for the control fruit. So then we ran some randomization tests to determine if this mean difference in observations was significantly larger or more positive than we would expect under the null hypothesis that the nymphs had no attraction to the treated fruit. So I'm going to show these results from both tests, the adult fed trial and the nymph fed trial. And remember, positive values indicate a preference for the treated fruit and negative values indicate a preference for the control fruit. And so here the histograms are sort of the expected mean differences under the null hypothesis of not having any attraction. And the blue line shows the actual observed value, so that actual average that we calculated. As you can see, for both the adult fed trial and the nymph fed trial, the bugs significantly preferred the treated fruit over the control fruit. So this was pretty clear evidence that Narnia ephemerata nymphs are attracted to conspecific cues left on cactus fruits, even in the absence of those conspecifics themselves. So they can actually tell when another bug's been feeding on a fruit, and they will seek out that fruit and spend more time on that fruit. So again, to return to the main aim of this experiment, our goal was to investigate conspecific attraction behavior in nymphs. And we found some evidence that juvenile herbivores may be using conspecific attraction during feeding to enhance their growth and survival. But it's still unclear if the behavior is actually adaptive for these juvenile animals, and if so, in what ways? Because like I said, there could be many reasons why this would be adaptive. Furthermore, this attraction behavior shown by the nymphs was directed at both adults and juvenile conspecifics, which was pretty interesting. So this may indicate that any adaptive value this behavior does have is probably not reliant on benefits provided specifically by adult conspecifics. So if they are benefiting from feeding facilitation, maybe it's from other juveniles and adults, or maybe there's some other reason that this behavior is beneficial. So overall, this project, we showed that nymphs do have attraction to cues left behind by conspecifics. And that takes us to aim number 4, which was to determine if nymphs actually seek out and reuse adult feeding sites. So this was really key because, of course, can't say that they're benefiting from feeding facilitation by adults through this mechanism of reusing the salivary sheaths if they're not actually going and feeding in the same spots as the adults. So we wanted to look at that behavior more specifically. So to test this, we allowed adult bugs to feed on a cactus fruit for an hour. Then we removed the adult and marked the feeding site. We also added a second mark on a different part of the fruit just to act as a control because we thought the pen we were using to mark the fruit might be attractive or repellent for the bugs. And then we placed a nymph on the fruit and recorded where it chose to feed, if it was in one of those marked sites or just somewhere else on the fruit. So what did we find? Well, throughout our experiment, we observed nymphs feeding 30 different times. And of those 30 feeding events, 2 were in the same sites as the adult fed in, and 2 were in these control sites, and the other 26 were outside either marked site. So overall, the nymphs didn't seem to seek out adult feeding sites. And so we didn't really have any evidence that they were using the strategy of gaining nutritional benefits through feeding facilitation. It seemed like they didn't really have any attraction to or avoidance of the marked sites at all. We actually tried to estimate the surface area of the fruit, and the area covered by the circles we marked was like approximately similar to what to 2 out of 30. So it seemed like they weren't attracted to or repelled by the marked sites at all. So for aim number 4, we had set out to determine if nymphs seek out and reuse adult feeding sites, and it turns out they do not. So for Narnia ephemerata, if the conspecific attraction behavior we observed earlier in juveniles is actually adaptive, it's based on this experiment, we'd say it's likely not due to feeding facilitation by adults. So returning to our list, you can say nymphs do not seek out and reuse adult feeding sites. Okay, and then that's takes us to our last project. So aim number 5. So here we wanted to test how the presence of adults actually impacts the growth and survival of developing nymphs. And so we wanted to directly examine whether proximity to adults provides fitness benefits to nymphs. And previously, if you remember from aim number 2, we had found that the presence of an adult did not help Leptoglossus innatus nymphs survive on nuts with hard shells. But we had not yet tested potential growth or survival benefits of developing with an adult present in Narnia femerata. And since Narnia only feeds on the Opuntia cactus, it's more of a specialist feeder. We thought they may have evolved different feeding strategies than a generalist feeder like Leptoglossus innatus, like I mentioned earlier. And while the last project suggested that Narnia nymphs do not seek out adult feeding sites, so that gave some evidence against the feeding facilitation hypothesis, we thought it was still possible they may benefit from feeding facilitation over a longer time period, such as their entire juvenile development. So we thought it was still worth testing potential fitness benefits of an adult throughout the nymph's development. So for this experiment, we raised bugs from eggs through adulthood in one of 2 treatments, either with an adult present or without an adult present. And the bugs were kept in cups with fruiting cactus and were initially in clutches of 10 to 20 eggs. And we tracked survival throughout the bugs' juvenile development. And once they got to adulthood, they were frozen and measured, just like in the first experiment. And we measured their pronotum width, which again is a proxy for body size. So here we're going to look at survival first and then body size. And we found no difference in the bug's probability of survival from hatching to adulthood. If anything, survival seemed to be slightly higher in the cups without adults than the cups with an adult, but the difference was not statistically significant. So this was the opposite of what we had predicted. We thought that they would have higher survival with an adult, and that was definitely not the case. And next, I'm going to show you the results from the body size comparisons. And again, I separated the male and female bugs because female bugs tend to be larger on average than male bugs. And again, there was no significant difference in body size at adulthood between bugs raised with or without an adult. And like with survival, there was a trend towards larger body sizes for bugs raised without an adult compared to with an adult. So the opposite of our prediction. But the difference was not statistically significant, although for the male bugs, it was pretty close to the p-value. So that indicates maybe more research should be done on that possibility of costs imposed by adults. So going back to our aim, we aim to test how adults impact the growth and survival of nymphs. And we found that adults do not provide fitness benefits for nymphs, at least in the controlled conditions of our experiment. That's key. The trend actually went towards greater survival and larger body sizes in the bugs raised without adults. So that suggests that if anything, adults might actually impose a cost on developing nymphs, possibly through competition. So more research is definitely needed to look into this possibility because our experiment didn't show anything definitively. And overall, we once again find that feeding facilitation by adults does not seem likely to help nymphs access nutrients, at least not to any measurable extent. However, adults may provide benefits to nymphs in other sort of more ecologically relevant contexts. So for example, the presence of an adult or of conspecifics in general may help nymphs avoid predation or find high quality fruits out in the field. So our experiment really was looking at potential benefits of an adult in this sort of laboratory context, which is devoid of predators. We gave them good food, et cetera. So it showed that feeding facilitation is likely not occurring, but there could be other reasons for that conspecific attraction behavior we saw earlier that are that would make that adaptive out in the field. So to finish out this list of the main results from my dissertation research, we can say that adults do not provide fitness benefits to nymphs, at least in controlled environments. So what can we take away from the list of results? Well, we found strong evidence of rostrum length plasticity in Leptoglossus zonatus, and this result aligns with previous research on Narnia femerata. However, this was the first study to show plasticity as a specific response to the thickness of a physical feeding barrier. And we also saw that structural plant defenses can seriously limit juvenile survival, even for generalist feeders like Leptoglossus zonatus. So this finding reinforces the importance of studying feeding abilities in the juvenile stage, rather than simply looking at adult animals. And we also found no evidence of feeding facilitation by adults in either Leptoglossus zonatus or Narnia femerata nymphs. So we super thoroughly investigated this hypothesized feeding strategy, both by looking at the nymph's behavior directly and by looking at potential fitness benefits in both species of bugs. And our results differ from those previous studies on the soapberry bugs, as well as that study with the other leaf-footed bug that feeds on the pine nuts. So more research is needed to understand the specific context in which feeding facilitation may actually occur, and what species and what types of host plants that may be occurring on. And lastly, we found that Narnia femerata nymphs are attracted to conspecific cues. So this was really interesting. This finding opens up interesting avenues for future research. For example, we still don't know if the behavior is adaptive, and if so, why and how. And does it only benefit young nymphs? So we didn't actually test for potential attraction in adults. So the conspecific attraction could be generally beneficial across all life stages of the bugs. And so these questions are all still unanswered and will hopefully inspire future research on these super cool bugs. So with that, I'd like to thank all my committee members, collaborators, and lab mates, as well as many undergraduate researchers who made this work possible. And thank you all very much for attending my talk. I'll be happy to take any questions.
I feel bad asking this because it's not like specifically about your research, but I was just more wondering in general how your focus on science writing maybe influences your research.
Oh gosh, that's an interesting question. I mean, to be honest, I had already completed most of the experiments by the time I started really getting into science writing for a general audience. But it definitely, I feel like writing up these studies and working on this presentation, having that science writing experience really made me think about how I was thinking about it, how I was talking about it, how I was communicating the ideas, and also having experience with scientific writing helped me with the, with the other types of writing. I feel like all different types of writing, there's a lot more in common than you think, even if you're, writing a scientific paper and then you think this is nothing like writing, a blog post. There's a lot more in common than you think. And I've gotten advice from advisors, mentors about to improve my scientific writing that then ended up being super applicable when writing for, Smithsonian Magazine and vice versa. So I think these ideas about how to communicate science really carry over. Yes.
So you mentioned that there were a couple of cases in Nymphed Trial species that more or less pointed to a fourth facilitation, but would you say that the consensus in general was to assume that there was facilitation in the field? How was this received?
So, and these studies have not been published yet. But yes, yes, the thing is, go back a couple slides.
Sam, would you mind repeating the question?
Oh,
It's hard to
yes.
hear.
Yes.
Yeah. I was wondering if at some point you mentioned that there were a couple of cases in which there was evidence of facilitation, but for a couple of species. But I was wondering if overall everyone was taking for granted that there was facilitation in the Nymphed Trial as a whole.
Yeah. So that's definitely, so no, this was, the main study showing evidence for feeding facilitation was the one from the soapberry bugs, so the Sensor 2021. And so that was published in 2021, obviously. And so this is sort of a newer idea. It's not been thoroughly studied. That's part of why I wanted to focus on it so much in my dissertation, is because it's not, the feeding facilitation hypothesis, the way I looked at it here, is not something that has been really well studied, is like well accepted or assumed to be going on. And so this is sort of, so pretty much first steps in terms of investigating this in more species. The other study, with the other leaf-footed bugs with the pine nuts, that all they showed was that 2 adults will use the same hole through the nut or through the shell. So they didn't, that was again like a first step at looking at that. They didn't look at whether that was actually had any benefits to them, or if it was just like convenience, it's a hole there, or if over a long term that would actually impact their survival. So it's really that Sensor 2021 study was the first one that I have found that really thoroughly examined, the shells with the cracked, seeds with the cracked shell, the early juveniles versus the older juveniles. That one was also, okay, this is a bit of a tangent, but it was interesting that with the soapberry bugs, the youngest juveniles got benefits from having the adults feed on the seed first, because it was easier for them to get through the shell. But older juveniles, they actually suffered a cost from the adults. And the explanation was through competition. So it went from facilitation to competition, which was super interesting. That by the time that they were older nymphs, they could feed through the shell just fine. And at that point, having an adult feed on the seed before them was just like, okay, they taken half the food, So yeah, that was, I would love to see more research into this in more species.
Thanks.
Mhm. Yes. No, so, well, okay, so the behavioral experiments were all with second or third instar nymphs. So again, first instars don't feed. So second instars, that's the earliest stage at which they begin feeding. I wanted to focus on using the smallest ones because I thought they would be most likely to get that benefit and most likely to show the behavior of, of conspecific attraction, seeking out adult feeding sites, et cetera. So I really focused on second and third instar nymphs for those behavioral studies. With the pecans, that study was looking through their whole juvenile development, and the Agar study that was looking at fourth instars through adults, well, fourth and fifth instars, the effects there in terms of plasticity. And again, there was there were some trade-offs because if in terms of what it could do in the experiment, because if I tried to have them from second instar through adulthood, they might all die at second instars if the agar barrier was too thick. But if the agar barrier was too thin, it might not have enough of an effect to really see the plasticity. So that's why here we just did the experiment from fourth instars to adults. Yeah, sorry, that was rambling. Did that answer your question?
And there's a question in the chat.
Oh, yes, chat question. Was there any correlation between exposure of conspecifics of any life stage and increase length of the rostrum? Might rostral growth / length be at least partially affected by exposure to competing conspecifics? Yes. So that, okay. That, I should have thrown that in here. So for, so for this study where we're raising the bugs with or without an adult, we also did take some other measurements, which I didn't put in here because there's already a lot in here. So I did also look at the rostrum length and at some leg lengths. It took a bunch of measurements. And there was no clear effect of the adult conspecific on the rostrum length, except for there was this one interaction effect that was significant. And it was just like the smallest males had a difference based on if the adult was present or not. But by the time they were at larger body sizes, this isn't going to help, but anyways, once they're at larger body sizes, the ones at larger body sizes did not have an effect. Again, males are smaller than females. So it was really just the smallest bugs that it seemed like there was an effect on their rostrum. So it was somewhat intriguing, but it was very hard to say anything definitive about that. I think we need like a follow-up study to really look at that in detail. Because yeah, there seemed to be something going on there, but it was hard to make conclusions about. Does that answer the question? Yes, cool. Thank you. That's a great question. Any other questions?
Yes. Thank you, Sam, for the research and sharing. It's really interesting. And also the research design is also interesting. It's not exactly a question, but maybe just trying to understand and thinking together. You mentioned that the species, like they feed from amount of group of species, right?
They feed from what?
I mean, amount of group, many species, plant species, right?
Oh, yeah.
And I mean, and I suppose maybe they have like different rates of nutrition, each one.
Mhm.
And maybe, I mean, just thinking, maybe when you did the laboratory food, I mean, in the Agar, I mean, I suppose you just had to choose one of the, I mean, one.
Right.
One recipe, right?
Mhm.
Maybe just one or two recipes. So maybe all these measures and dynamic of size and even the size, the growth of the mouth part could also be, I mean, in a different situation depending on the diversity of feeding.
Right. Yeah. So yes, no, that's a great question about the sort of like diversity of their diet and what nutrients are getting. Sorry, I'm going to go back to that. So this is the Agar experiment with these agar plates and with the sunflower seeds. So they really, so actually in their communal housing, they got like tomatoes and snap peas, I think. Oh yes, it says it on there, tomatoes and snap peas. And then it's once they're fourth instars, they're transferred to this diet that was like pretty much, they were only eating sunflower seeds. And so, yeah, so there could be like in the wild, more nuance about like what nutrients they get when, and that could affect their development. But a key thing to keep in mind is that even though they are generalists at a species level, at an individual bug level or at a population level, they're often just eating the same host plant throughout their life. So remember, the nymphs don't have developed wings, so they can't fly. So if their eggs are laid on a tomato plant, they're generally like eating that tomato throughout their life, unless then, okay, the tomato stops making fruit. Okay, maybe they need to move to the pepper plant that's like right next to the tomato. I don't actually have, I can't give you some numbers or anything about how far they typically disperse in different locations, but they can't just go flying anywhere looking for other food. And it's not like they're just moving around day to day saying, oh, now I want this food, now I want this food. So exactly, yeah. And same thing with the pecans. I mean, that's the same species. But when we think about these nymphs developing especially like in an agricultural context, they're likely not going to have other plants around that are good host plants, because if in that area, people are just growing pecans, then that's what they have. And so if the nymphs can't survive on the pecans, then they're just going to die. So yeah, it's a great question. Yes.
Can you show your objectives again?
Oh, yeah.
Do you have any hypothesis on the difference in effect of only testing a single conspecific versus multiple conspecifics in the experiments?
Yeah, that's a great question. Yeah, because mostly I was just looking at like that single conspecific, because thinking of they're creating that a certain hole in the shell or the salivary sheaths, then I was mainly looking at the effect of none versus one, or none versus any. Haven't really thought much about sort of group dynamics, mainly because in the context of these experiments, overcrowding them in the cups can cause mortality. But out in the field, they're often sort of like aggregating in groups to some degree. So that is a good question. Often they aggregate more as nymphs, and there might just be like one or two adults near a bunch of nymphs, which is mainly why I was thinking about it as a none versus any in terms of the adult effect. Yeah, I don't have a very good answer to your question, but it is an interesting thought. Yes, don't have any hypotheses off the top of my head.
And can you show your expectations on the survival plot for the pecan? Maybe I missed it, but in your thinking, was there a reason that the no shell adult would exhibit greater survival than with the nymph? I mean, it looks pretty minor if that's actually, or did you say they were
Like the orange line versus the red dashed line? Or-
blue, green?
Oh, you mean differences between the blue? Oh, no. Yeah, I expected no differences. I was just.
Okay, I just missed that then.
On
Okay,
top
yeah,
of each
thanks.
other. Yeah, so no, there was no expectations of differences between no shell plus adult and no shell plus nymph, because we had no hypothesis about the conspecific life stage affecting the nymphs in any way without the shell present.
Well, thank you so much. Is mine open? Oh yeah, okay. Yes, so thank you so much. That was awesome. I'm getting comments here, direct messages about how interesting this is. And so thank you so much for sharing your research with us. And so for everybody on Zoom and here, obviously if you have questions for me, and then also Sam, I believe your email is on the flyer that we sent out. So if you have additional questions for Sam, you can always use that. And otherwise, I hope you all have a great week. Thank you.
Thank you all for coming.